Today I am talking about how an experiment is designed in medical science. I will use application of quantitative PCR or RT-PCR for designing an experiment in medical science. PCR, as I have mentioned earlier in videos, is used to amplify a specific segment of DNA, generating millions of copies of a target sequence, while the RT-PCR or quantitative PCR also amplifies DNA like PCR, but in addition to amplifying, it quantifies the DNA in real time as the amplification occurs. And I have also mentioned in my earlier videos that when we isolate RNA from our samples, we make a complementary DNA from that RNA. So in other words, we can say that quantitative PCR can be used for the amplification of a complementary DNA, and it also quantifies the complementary DNA, which is actually a gene, in real time as the amplification occurs. Okay, so how a discovery is made I cannot say like that. Rather, I can say that how an experiment is designed and how we can use gene expression analysis or quantitative PCR or real-time PCR, which are same thing, for making a discovery or for designing an experiment. Okay, so first of all, we will go through the literature. For example, I studied the literature and I find that there is pituitary gland and there are stem cells in the pituitary glands which have been discovered earlier. For example, these thyrotrophs, which are the cells in the pituitary gland, and they are secreting some factors. The name of these factors are factor A. And then I find another paper where the scientist has reported that the thyroid gland is releasing the precursor of thyroxine, which is thyroglobulin. That is the second group of scientists who have came with this paper. And I also find that actually this thyroglobulin is released by the follicle cells of thyroxine. And I also studied a paper where I found that on the surface of follicle cells of thyroid gland, there are receptors, and one of the scientists has reported that these receptors have been found to be attached with the factor A, which is actually coming from pituitary gland. Now, these are the information which actually we have, but it has not been discovered that what is the actual role of this factor A inside the body. And now, based on this information, we can design an experiment to find out the role of this factor A, the physiologic role of this factor A in human body. We can make different hypotheses. One of the hypotheses we can make is there any role of factor A released from the pituitary gland in the secretion of thyroglobulin? So based on this information, I can make a hypothesis that already so many scientists have discovered so many things about the physiological mechanisms related to pituitary gland and thyroxine. And I am coming with a hypothesis that this factor A, which has been found attached to a receptor in the follicle cells of the thyroid gland after being released from the pituitary gland, may have a role in the secretion of thyroglobulin, which are the precursors of thyroxine. For this hypothesis, I can design an experiment. I will take the cells, follicle cells from the thyroid gland, one group of cells where I have not done any mutation. These are the wild type follicle cells, which have the receptors on their surface, and that receptors can bind with the factor A, which is a chemical released by the pituitary gland, and we don't know its actual function yet. And now I will mutate this receptor on the surface of the follicle cells. Now this group of cells where I have mutated the receptor on their surface is unable to attach with the factor A, which is coming from the follicle cells, which is coming from the thyroid trough cells of the pituitary gland. Now I am looking that either these cells are capable of secreting thyroglobulin, or either these cells are capable of secreting thyroglobulin, which is a protein and a precursor of thyroxine. So this is now my experiment. I have two groups of cells, one wild type cells, one mutated cells, where I have mutated the receptors for the factor A. And my hypothesis is that actually this factor A, which is now unable to bind with the follicle cells, is responsible for the release of thyroglobulin. Now my expected result is that these cells where the receptor is intact can bind with the factor A and is capable of secreting thyroglobulin, while these cells where I have mutated the receptor for the factor A, they will not express the thyroglobulin gene, or they will not make the thyroglobulin protein, because my hypothesis is that actually the thyroglobulin secretion is controlled by the factor A, which is coming from the pituitary gland, thyroid trough cells of pituitary gland, and I have disrupted the link between the factor A and the follicle cells. That is why thyroglobulin will not be released. That is my hypothesis. So to check this hypothesis, either to prove or to nullify this hypothesis, I will collect RNA from this sample and this sample. And how we will collect RNA, that I have already discussed in my earlier videos. And then I will make a cDNA from this sample as well as from this sample. And I have also discussed that how cDNA is made from the RNA. Okay, so now I have collected RNA from the wild type cells, from these cells, as well as from the mutant cells, and I have made cDNA from the RNA. And now I am going to check whether in this mutant sample there is a thyroglobulin gene which is expressed or not. In other words, I can say that either the thyroglobulin RNA is found in this sample, and I am expecting that it has to be in this sample because I have not done any genetic manipulation in these cells. These are wild type cells. So for that, what I will do is I will take a an append draft tube, 1.5 milliliter append draft tube, and I will put here cyber green, which is a fluorescent dye, and when the cDNA or complementary DNA or your gene of interest will amplify, the cyber green will be attached with that gene of interest, and it will give fluorescence, and that will be detected in the pharma graph, and from that we can quantify either the complementary DNA or your gene of interest is in this sample or not. So I will put 60 microliter of cyber green in this tube, and I will put 6 microliter of forward primers for the thyroglobulin in this tube, and I will put 6 microliter of reverse primer for the thyroglobulin in this tube. The reason is that I am going to quantify and detect the thyroglobulin gene. That is why I am using forward and reverse primer for the thyroglobulin gene. And I will not put this DNA or the complementary DNA from these two here. I will put it separate, but I will put 36 microliter of water in this tube. And after that, I will mix it and vertex this master mix. And from this master mix, I will put around about 17 or 18 microliter in each of these wells. This is a 96 well plate, I think, and I will put 17 or 18 microliter of this master mix, which has cyber green, forward and reverse primer, and water, but I have not put DNA there. So I will put 17 to 18 microliter of this master mix in each of these 6 wells. After that, from this sample, I will put 2 microliter of cDNA here, and then again another 2 microliter here, another 2 microliter here. That 2 microliter may have like 500 nanogram of complementary DNA. And then I will put 2 microgram of complementary DNA from this sample to here. So now I have a triplicate of each sample, and in these samples, the sample of the complementary DNA have been mixed with the cyber green forward and reverse primer for thyroglobulin gene as well as water. Then I will centrifuge it briefly at 1200 RPM for 1 minute, and then I will put this. Now I have like these 3 wells have my wild type DNA, and these 3 wells has mutant DNA, which is actually a complementary DNA of the RNA. So I will centrifuge it, and after that I will put it in the quantitative PCR machine, and I will start this software which I am using, 7500 software, which is the name of the software which is used in this PCR machine. And then I will design a new experiment here, name my experiment in this desktop, and I will select fast 96 wells because I'm not, you can make a different type of, you can design different type of experiments, but I am just interested in getting just graphs, either the gene is expressed or not. So that's why I will select this setting. I will select delta delta CT, and I am using cyber green, so I will select cyber green, many of the scientists are using Tegman technique. That's also great technique, but I usually use CyberGreen, and then I will select fast run because it will be completed within 45 minutes. In slow run, it will take 2 hours, and then I will define my target. Defining target mean I will say, I will write here that these first three wells have wild type DNA, and the 4 to 6 have mutant, the complementary DNA from the mutant sample, and then I will also mention that the gene I am amplifying in these samples is a thyroglobulin gene. And after that, I will start run, and so that the quantitative PCR will start running the experiment. And after 10 to 15 minutes, the graph starts appearing here for the different plates or the different wells which we have put there, and we are interested to see whether the sample there has the gene expression or not. So after 45 minutes, when I will go there and I will see if my hypothesis will work, where my hypothesis was if I will mutate this receptor, the thyroglobulin gene will not express. So I am expecting now, theoretically, I am expecting that in these 3 wells, I will get this type of graph where the fluorescence and copies per reactions, which is also called the CT value, it will give me this type of graph, which means that my gene of interest, which is thyroglobulin gene, is amplifying here. And theoretically, for these 3 replicate, my hypothesis is that I will not get this type of amplification graph, because I am expecting that when I mutate the receptor on the surface of the follicle cells of thyroid gland, the thyroglobulin is not expressed. And if the thyroglobulin will not express, I will not get this type of curve or graph. And after that, I will bring these results and I will go back to my hypothesis, and I will see that now, for example, this was my design. I had designed an experiment where I had mutated this receptor on the surface of the follicle cells, and earlier reports have shown that this receptor is actually attached with a factor which is coming from the thyrotrough cells of the pituitary gland. But the function of this factor is not yet known, and for that I have designed an experiment. And my hypothesis was that when this factor is stopped to interact with the follicle cells, it will not release, the follicle cells are unable to release thyroglobulin. So with this hypothesis, when I see that the RNA which was coming from these cells are amplifying thyroglobulin, this gene, and these cells are not amplifying this thyroglobulin. So what does it mean? It means that actually everything was fine. The factors were there, these factors which are coming from the pituitary gland, these were also treated with these cells, and these cells were also treated with these factors which are coming from the pituitary gland. Here the receptor was intact. Here the only difference was the receptor were like deleted, they were mutated. So due to the mutation of this receptor, this factor was unable to attach with the follicle cells, and hence the thyroglobulin was not formed or the thyroxine was not formed from the thyroglobulin. So what does it mean? It means that the factor A, which is coming from the pituitary gland, it is actually responsible for the release of the thyroxine. And this factor A, we can name it thyroid stimulating hormone. So I think that when the thyroid stimulating hormone was discovered, I expect at that time, if the quantitative PCR was there, this experiment must have been done, that let's delete the receptors for this factor on the surface of follicle cells and treat the cells, the wild type cells and the mutated cells, receptor mutated cells with this factor A, and see either the thyroxine is formed or not. And if the thyroxine is not formed, it means that this factor, which was earlier found, but its function was unknown, that is actually thyroid stimulating hormone. So that was an example. You can design in this way. You can design your experiments in this way. You will go through the literature and you will find some reported products in the extracellular matrix or in the blood or inside the cells, and to find out the function of that product or that protein, you can design this type of experiment, and you can use a quantitative PCR to address this type of questions. So that was all about how experiment is designed and how quantitative PCR can be used for addressing any biological question. Thank you so much.